Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. I'm super excited to have Professor Hemal Patel of University of California, San Diego with me. Professor Hemal Patel has been at UCSD for close to 20 years now. He started his career with a PhD in pharmacology. So that's the study of drugs and its interaction with living systems. And he is a pioneer in the study of mitochondria and how cell membranes also talk to the outside world. And he's one of very few scientists who can talk about biology and philosophy at the same time, because he also had a double major in biology and philosophy. We scientists reinvent ourselves throughout our lifespan, and Hemal has reinvented himself. And from pharmacology, or while doing pharmacology research still, he has also pioneered the scientific study of meditation. And so that's why I feel this will be an exciting session, because very few of us straddle that domain where we study both drugs and mindfulness or meditation on our health. So welcome, Hemal.
Good to see you.
Yeah, so it's always a curiosity. Means we start something in high school, curious about science, and then look at yourselves.
Yeah.
You are here. So what are some of the turning points, inflection points over the last 20, 30 years that actually brought you to
Where I am, yeah.
where you are?
It's an interesting question. You know, my oldest is starting college, and so it brings you back to when I was in college and thinking about that path. And high school was, I think, a transformative time for me. I was born in India, moved to the US when I was 6 years old. Grew up mostly in the Midwest, right? And so the idea was that's where culturally Indians sort of lived, in that Chicago area. My parents moved into the suburbs, so there were very few Indians. And so my surroundings was mostly white. I think my sister and I were the only brown kids in the school. And so that shapes you in unique ways. There's challenges where people are completely accepting, and then you get the extreme where they don't know who you are and you get picked on because you look different from everyone. Mm-hmm. And so I think that for me built a very thick skin, and I'd always been this kid who pushed back, right? I was not someone who shied away. And I think that shaped a lot of my future and where I ended up. As a brown kid in a very sort of a family that looks for education as a key. Most Indian families have that.
Yeah.
At that time, there were two choices you had. You could either become a lawyer or a doctor, right? And
there was no engineer?
Engineer was not a thing at that time, at least in our community. And you had to be the right kind of doctor. A PhD was unheard of, right? An MD was the real doctor kind of thing. And so that's what I grew up envisioning. I was always very good at math and science. Those things came naturally to me. High school, I didn't really have to study, right? Natural abilities to just process math and internalize stuff. We have this joking thing in the family. So I met my wife in high school, who's not Indian. And so that was a huge defining point for me as well, right? Being in this sort of mixed marriage where we had to navigate sort of her family, my family, and everyone's dynamic of what they want. But I was valedictorian, and she was also very highly ranked in the class. And then she had a brother, I had a sister. And so her brother was valedictorian a few years later, and then my sister was valedictorian a few years after that. And the joke is she's the only one that wasn't a valedictorian. But out of all of us, she's probably the smartest, right? She has this amazing ability to just learn and retain information and do things. And so this shaped a lot of who and what I was. So she grew up as a Catholic, and I had no concept of Western theology. And so then when we made independent choices, we ended up at the same college. And the idea was that I would go in as a biology major thinking that this would lead to this path to becoming an MD. And then first semester in college, I was at a liberal arts school. And so you had to dabble in all of the other areas to get a degree. And so I thought, I've never taken philosophy. So I took philosophy. I just fell in love with the course. And it's always a teacher that ignites that passion. Topic was interesting, but she was this crazy, amazing individual who talked about existentialism and all these crazy experiences that she had. And Natalie Alexander shaped my path. And so I did very well in her course. And so she said, you should take my next class. And so when they encourage you, yeah, all right. And so then she's like, you should minor in philosophy, right? You only had to take 5 classes to do that. And then I did that. And she's like, well, you should think about majoring in philosophy as well. And so it intertwined really nicely with the stuff I was working on. And it was an opportunity to explore other aspects of my life that I never had. And so my girlfriend at the time and wife was Catholic with this Western theology. And so I immersed myself in the philosophy religion aspect, really the religious aspect. Growing up as a Hindu, I knew nothing about Hinduism other than you prayed to a temple in your house once in a while, especially when you had a test. You'd go, you'd make sure you got a good grade on the test. You light a candle, and that was sort of the extent of it. And then all the superstitious things that go along with drinking this during this time of year and all those things, right? I never really understood the foundation of Hinduism. And so I explored that Eastern aspect as well. And it was really
in college.
in college. And so it was an awakening to actually learn about this religion that was mine, but never really was. And so I really learned religious aspects, Eastern, Western theology, a lot of philosophy kinds of things. And at the same time, it balanced with biology. The biology thing was something that just, again, was natural and easy to do. Science wasn't really hard. I loved organic chemistry. It was one of these classes where I could see things in my head, and it was good for that, right? So fast forward to the end of college, and we're trying to make decisions of what to do. And my wife, Jenny, decides she's going to go to medical school. And it was something that she'd always, either she becomes a teacher or a pediatrician. And so she leaned towards the pediatrician thing. And then I made the decision to not go to med school. And I worked at Abbott Labs for a couple months over the summer and then 6 months, and then decided to do a PhD, which no one in the family understood. And so this is someone who had this intense intention that I was going to be something in the future that was around medicine. And now I'm the only one in the family as the child who's not an MD. All the rest of them are physicians. And I'm the only one that's a PhD. And I look back and I think it was all of these unique experiences living in this culture where I struggled, but also had a unique look that no one else had, having this early person I interacted with who became my wife, going to undergrad where I really expanded in this unique kind of way and became a liberally educated individual from the perspective of really high-end classes in things that were not biology that then really shaped me as a scientist. It allows you to think way outside the box, and you're not pigeonholed into a particular idea of I'm a neurologist or I'm a neuroscientist or a cardiac physiologist or something like that, right?
Yeah.
Ask a question and answer it. And that's what you're taught how to do.
But I guess if we go back to your high school,
Yeah.
you mentioned that you went to Midwest and suburban living. You're the brown kid who was outlier. And I think in those situations, you want some kind of acceptance.
Yeah.
And do you think, I don't know whether your wife at that time was your girlfriend or just a friend happens to be a girl,
Yeah.
whether her accepting you
Yeah.
was an inflection point, and whether she was discussing more theology or philosophy with you or not. Do you think that was the inflection point? And I was also curious, how did your conservative most Hindu families are conservative in that sense, and how did they accept her?
That was a challenge, right? So We were boyfriend and girlfriend. We knew each other because we were in all the advanced classes, and so you have someone in your view, but you never really think along. We were friends, right? And I asked her. I didn't get the nerve to ask her out until senior prom. So my last year, it was do or die kind of thing, and so I did, and she said yes. And she had had the same kind of feelings about me, but just never really expressed them in that kind of way throughout that time. And I think there is some element of acceptance that goes into that. Not that I needed that, but it validates that. And I think when I look back, for a long time, I shied away from my Hindu culture, right? Because it was something that ostracized you away from being accepted and being around. And so we had two personalities. At home, you were Indian, and at school, you were as white as you could be, right? And luckily for me, when I grew up, I learned English as a first language in India. My parents knew they were going to come to the States, and so they pre-planned for all of this. And so I had no accent, and so I didn't stand out from that. Other than my skin color, I didn't look or I didn't sound any different than anyone else. And so that I think was an advantage. The other aspect is you get pigeonholed as a something. And so my something was I was the nerd, right? I was the smart kid. But then I was also involved in sports and things. I played tennis, I ran track, I did things that kept you out of that nerd role kind of thing. And so when we started dating, sort of the funny thing is, so we were really nerdy kids. And I had never dated a girl, right? This is the first date I was on. I asked her to, Schindler's List was out. And we're like, do you want to go watch Schindler's List? And so that was our first date. We went to watch this really serious movie about the Holocaust. And we had this intense discussion afterwards about, and that's how our lives have always been, right? We have these really close interactions where we talk about stuff in a very deep kind of way. And so she knew nothing about Hinduism either. And so our summer project, that summer before we went away to college, was to read the Bible from cover to cover and talk about each of the books. The Old Testament was a pain in the ass to get through. I mean, Leviticus and Numbers, that's just as boring as you can make it. But it's important to learn that progression. And then she wanted to understand Hindu culture. And so we read the Bhagavad Gita, the Ramayana, and the Upanishads, right? These
Upanishads too.
Yes.
Wow.
And the abbreviated versions of all of these.
Because that's pretty long.
It's long. And so it was this immersion in this kind of cultural exchange. Our parents didn't like this at all, right? Her mom, who was Catholic and raised the kids Catholic, was very upset with this idea that her daughter was not dating someone who's white and Catholic. I don't know if the white mattered, but the Catholic part did, which parents tie onto. For my parents, my dad was really fixated on this idea that I had to marry someone who was Indian because that's how it is, right? Their marriage was arranged and that was the expectation. And it's sort of an odd expectation that you leave the country you're from where there's all these Indian people that you can interact with. You put us in the middle of nowhere, Chicago, and there are no Indians around, right? I mean, they're around, but you're not
your next door
constantly.
Indian is 20 miles away.
Right. And so it was inevitable. And I think that inevitably came out. I think the one person that really understood and really was one of the driving forces in this was my mom. And so my mom was always, parents are supposed to have favorites, but I think I was her favorite. And they have this, she just had this personality that was just completely accepting. And it comes from your parents. And so her parents raised me when I was younger, which is the typical thing you happen when you're in India is your parents are working, your grandparents raise you. And my grandfather was just this worldly, amazing guy. And he had had the worst hand dealt to him. And he was just one of these guys who just let it brush off and would be successful when he'd go do something else. And so he had emigrated the family to Africa. So they're in Tanzania, owned a cashew factory, and quite wealthy, right? They had just servants and learned Swahili. And so all four kids were raised in that environment. When all the dictators came into power, the Indians got kicked out. And so they took his factory and he had to go back to India. And so my oldest uncle on my mom's side went to London, became a pharmacist, and did very well. And then three younger kids went back to India. But that world environment that he entertained, he taught his children to think this way. And so education was very important for him, and just this kind, gentle being, right? And I have these, there's very few people I have old memories of, and him, I have these old memories of. I was, and this is, you're, as a scientist, your brain never leaves the science background. A couple of years ago, when I was in Paul Insel's lab and we were talking about some stuff, I was walking through the VA hospital, and I had this crazy, weird, aseptic smell of hospital, and it brought back like a 40-year-old memory of my grandfather riding on his handlebars of his bike in India. I could feel the sun on my skin, and it was just this intense memory. And I think there's a pharmacology that gets entrenched, right? And I just activated the right olfactory sensors that brought that intense memory back. And so there are people like that that shape you. And I think he shaped my mom. And I think she passed this on, and she was the only one that sort of was reasonable in all of this. And I think that's why my wife and I ended up together. And now I think I should know, right? It's 27 years now in August that we'll be married. And it's just, if we didn't have her, it would have never happened. And so I think that was a very important figure in our lives, I think, growing up.
But I guess that's also it. That's the origin of thinking outside the box.
Yeah.
Because you went outside this Hindu conservative thinking.
Yeah.
And having someone to support, and that's your mom, and that also you're influenced by your grandfather.
Yeah.
So I think in many of our lives, there are these stories where there are events that happened maybe 100 years ago.
Yeah.
That indirectly influence us.
They do, right? This whole butterfly effect kind of thing.
Yeah. So then in college, you go on to do your PhD in pharmacology.
Yeah.
Why pharmacology out of all of this?
Again, it's people, right? People touch you in unique ways. So my first lab I ever worked in was a circadian lab. It was this guy, Daniel Janik, and he was working on the suprachiasmatic nucleus, how it regulates motivational behavior and changes in circadian clock. And so we were working with Syrian hamsters. And so the project that I did for the summer in his lab was we were using opioids as a way to reset the clock. And so the idea is if there's a reward opioid system going on in the brain that controls some elements of the SNC, could you reset it by giving them naloxone at a specific time? So you acclimate them to complete darkness. And then at that time, we had this monocular camera.
Infrared.
Yeah, and so you're wearing this stupid helmet with this camera. You're going into this room. It's like the hand of God coming to pick up this thing. And we had this chain because they have really sharp teeth.
Yeah.
So you grab this thing and you inject it, and sure enough, you'd reset their clock by 3 hours by giving them the specific timed injection of naloxone. I remember...
So, just for regular people, what is this drug and what does it do?
So naloxone is a non-specific opioid antagonist. So there's multiple opioid receptors, and naloxone will stop working at those, will stop regular opioids from working at that receptor, all of them, right? Mu, delta opioid kind of thing. So non-specific antagonist. And so I remember this summer, it was me and this other summer student as part of this program. And Jen went into the room and she lost one of the hamsters. She's like, I can't find it. And so we changed outfits and I had this helmet on, looking around, moving everything in this room, probably disrupting the whole experiment. This stupid hamster had wedged itself up above on the thing, and it was gripping the wall. And so we finally figured out where he was, got him out, and put him in this thing. But that was the first thing. And so how did I end up in pharmacology? So Dan Janik left, and I had to find another mentor on campus. And so I found this amazing person, a scientist. She was a microbiologist, Cynthia Cooper. that taught the microbiology courses, but also ran a cell biology lab. And so I started working in her lab, and we were looking at purinergic signaling. So we're looking at how cancer cells release ATP and how that signaling is involved. And when it was time for me to think about what I wanted to do for grad school, I went and asked, well, what should I do? And she's like, oh, you should do pharmacology and toxicology. That's what I did, right? That's what I did some work in. And she said, if you do that, you can work in any space you want, right? As long as you learn the nuts and bolts basic, you can be a cell biologist, you can be a physiologist, you could do whatever branch of science that you want to do. And so at that time, they didn't have these blanket large programs. There was a program in pharmacology and toxicology. And so my timing was a little off, right? I always thought I was going to go to med school. And so I'd applied and then decided not to last minute. And so my girlfriend, who was going to be my wife soon, had gotten into med school. So I was waiting to see where she would get into med school. And then that was where I was going to apply to grad school. And so she had gotten into a number of med schools and decided to go to the Medical College of Wisconsin. And so then that was the only grad school I applied to. And I had pretty good GRE scores, and my grades were really good from undergrad. And so they took me off cycle, right? So I didn't have to wait a full year. I started in January. And because of this pharmacology, toxicology thing, that's the program I applied to and gotten in. And I worked and this was the time when you could do a 6-month rotation if you wanted to in a lab. There were no limits to anything. And so my first rotation was in a neuropharmacology lab, and we were looking at RAN binding protein and its role in nuclear transport, which Blobel eventually went on to win the Nobel Prize for. And so this guy that I was in was looking at eye systems, and it is a great model for this because it recapitulates very quickly. And so we do lots of work with cow eyes and sheep eyes and things like that. And so it was ultimately not the place I ended up. It was a lab where he was a very strong personality in terms of a PI, which can be okay. But I could see very quickly that I was going to learn a lot of stuff, but I was never going to be able to learn to think on my own.
Oh,
right? Which is fine. I think that's how most labs teach graduate students. They teach them to be really good technicians. Not what I wanted for my future career, right? I wanted to do something, like become a professor or something. So at the same time in the department was this older gentleman PhD, who ran, everyone knew him. He was just one of these joy-filled individuals. And so I ended up asking Garrett if I could do a rotation in his lab. And he said, well, I don't really take on more than one grad student or postdoc at a time. My other kid's leaving soon. So yeah, I'll let you do a rotation. But unless we can figure out money, I don't know, right? So you can do a rotation, maybe you have to do a third one. We had a blast in the lab, right? It was just an amazing lab to be in. He's the kind of guy who has this personality where he's had the same technician for 20 plus years. You know that there's something magical about that, right? Who can retain someone for that much time because they just love to work with him? And so Ryan Fryer was leaving at the time. So Ryan had decided to go do a postdoc at Harvard. And then he ultimately ended up in industry, and we're still in contact. And there's this family that he creates in his lab. So people that trained in his lab 30 years ago, I'm good friends with because there's this network, right, that's built. And so Ryan had left. I was sort of taking on his projects. He'd just gotten an R01, and so he had funding to take me on as a grad student. It was a time when I just expanded in a dramatic kind of way. We had to take 2 years of classes, which going through that as a student and now thinking about it as a professor, I think that's way too much time that people spend on that, right?
in grad school.
I think you learn to read very young. And if you're going to be a scientist, you should give them the honor and ability to say, if you have a problem that you don't understand, go find a book at the library and pick it up and read and learn, right? I think that's the skill that needs to be sort of taught, not the nuts and bolts. It should be expected that you know cell biology. And if you don't, you're going to go figure it out on your own because it's important. And so anyway, we had a lot of courses. And so I got to learn some really cool stuff. And it's one of the only departments in the US that has a toxicology component as well. And that I've loved learning about P450 enzymes and how we get rid of toxins and things like that. It's a huge component of this. And so I ended up in this lab where Garrett and his wife Carol didn't have kids, and so everyone who was in their lab was their child, right? And he poured his complete scientific energy into you. And he had these two technicians that were always in the lab, so they could do everything that all the grad students had trained them. And so the task was that you learn how to master the surgeries, all the things you want to learn how to do. Once that's done, your job is to teach and tell the technicians in the lab what to do. So you basically become a PI as a graduate student. It was that freedom was amazing, and he'd protect half of your day to read and write, and he'd expect you to spend that time in the office reading and writing. And so every one of his students is heavily published. I was the baby of all of them, right? I only published 19 papers as a grad student and only 14 as a first author.
Only 40.
I was the slacker. Everyone else had like 26 papers. But I also did this in like 3 years. And so he knew that Jenny was in med school and that when she matched and left, I was going to be left alone or I had to go with her. And so he knew that time frame. And so he helped really sort of focus on graduating in that time. And he'd been experienced with this. Most of the students, a lot of students that came through his lab were MD PhDs. And so he turned them around in 3 years. And so it was just this amazing lab to be in where you now just expansively ask big questions. And this is where my interest in mitochondria originally started. So the project that I was working on is Garrett and a student of his about 10 years before I started had discovered that opioids exist in the heart, that the heart is an endocrine organ that makes endogenous opioids. And these get released, and there's receptors that then bind to these opioid receptors, opioids, and they create a cardioprotective intervention. And protecting the heart has always been a stress event, right? And so there are always GI coupled proteins that then create this effect. So it's these inhibitory G proteins that things like opioids will bind to, and they shut a system down.
Yeah, so there's a little bit more technical here. So let's go back to opioids. When you think of opioids, we are always thinking of
Morphine,
crisis,
right?
And morphine.
Yeah.
So what you're saying is the heart produces its own morphine-like molecule.
Yep.
And that go and act through the lock and key kind of system.
Yep.
So the lock is the G protein coupled receptor that triggers downstream signaling that tells the cell what to do.
Yeah.
So why heart produces this opioid and why these are important for protecting the
there's the ventricular myocyte has these endogenous things, and I think one of the things that they're necessary for, the heart is an organ that can die, right? And so it needs to create some endogenous mechanisms to preserve its survival in these situations where it's having a heart attack and it's losing nutrients and oxygen and those sorts of things. And opioids are a molecule that does this. And so I would think evolutionarily that this is one of the things that the heart had sort of come up with as a way to protect itself, right?
So only when the heart is under stress, that's when...
I think that's when we think that these opioids get released, right?
Released more.
Yeah. And adenosine and other things work in a similar kind of way. So there's lots of chemicals that are endogenous that create this natural protective ability. And so this was discovered in the heart. I used to give these talks on cardioprotection to the grad students, and the concept of preconditioning in the heart was discovered in the late 80s by this Murray, Jennings, and Reimer. That paper has been cited thousands of times. And Chuck Murray, who was a faculty member at the University of Washington, was a medical student at the time when they made this discovery. And it was, what they did is they had this model where they were trying to create a lot of injury. And so what they did is they created small amounts of injury, thinking that a small amount of ischemia reperfusion, this loss of blood and nutrients to the heart, would create this massive injury. Paradoxically, it turned out that created a protective element. So now the heart could learn what's going to happen when it has a massive heart attack, and the heart's magically preserved. The tissue doesn't die, right?
So how do they do that? So they take a mouse or a rat and then tie up the blood vessels.
Yep.
What is the procedure? How do
So I used to do this in a rat, right? You go in, you open they're artificially ventilated, so you put a tube into their trach, you ventilate them with the machine, you open up the chest cavity, and then you have this suture that you put around the coronary bundle, and then you essentially clamp down on that, and so it restricts blood flow to the heart, and the heart becomes this sort of grayish color, and there's no flow to that. And so you create, basically, when you create that bundle, you're restricting blood flow to specific parts of the left ventricle. And so then you have a region that's at risk, and then you reperfuse. And so that's the other aspect, is the injury usually happens during reperfusion. So the heart can live under ischemic conditions for a little bit, right? Not forever, but you can live under ischemia for a while. But when you do that, everything starts to shut down. The mitochondria are going into this hibernative kind of like state because they don't have the resources they need. The injury happens when you reperfuse, when you bring the blood back into the heart. It's that huge amount of oxygen that returns that causes essentially a free radical crisis, right? The mitochondria are going haywire. They're making a lot of these reactive oxygen species, which then start damaging proteins, lipids, DNA, RNA, and you get this massive injury that happens.
So that's what also happens in heart attack, because blood flow to some part of the heart is blocked.
Yeah. And so this was this magical discovery, right? That if you just do small ischemia reperfusion, let that heart know what the stressor is, the heart doesn't die. And so since then, people have been trying to find a magic bullet, right? A drug that does this. And there's hundreds of compounds and protein targets and other things that people have discovered. And the opioid receptor was one of these. And so this is what the series of thesis was around. And so my thesis focused on what we call the second window of protection. So it turns out that there's an immediate protection that the heart has, but then which is through just modifications of things that are already there. But then there's this long-lived effect that it wanes and then comes back a couple days later. And we think that second phase is really new genes that are being transcribed by that stressor and proteins that are being translated, and you see this massive effect long term. So it's to define that particular aspect. And it turns out mitochondria become a core feature of that. They're protected in this dynamic, unique kind of way. And so that sort of launched, right? It was this amazing environment to be in, and you could just sort of think way outside the box and just ask these crazy, amazing questions. So it was a great lab to learn in.
Yeah, so from there, is there any new drug that actually...
That's the sad part. So over 35 years of people working on ischemic preconditioning, nothing has come to market. And you think about that as a researcher, right? The NIH has dumped hundreds of millions of dollars into this idea. So why hasn't anything come out of this? And now that I think back, it's that everyone's been using the wrong models. Everyone uses young, healthy animals in heart disease research.
Yeah.
Young people don't have heart disease, right? And so I think the problem is that most scientists will not make a complicated model because it takes time and a lot of money, and there just aren't the resources to do it. And so I think if we really want to change drug development and moving things to the next level, we need to create human complicated models in animals that we don't have access to right now, right?
Yeah.
Make them as complicated as you can to resemble the human condition. Yeah. And then there's always the problem, right? Does a rat or a mouse translate to a human? Human heart, if it's well developed and exercise, probably beats 60, 70 beats a minute. Mouse heart, 700 beats a minute. It's 10 times faster. So the physiology is dynamically different between those two, right? And to correlate it, it's hard.
Yeah. But that took you to the direction of mitochondria.
It did.
Yeah. And then how do we understand our mitochondria? Like, how can I figure out how are my mitochondria?
Yeah. I mean, that's I think a loaded question, right? Mitochondria are, I've been thinking about this a lot recently. We're working on this paper right now where it ties the mind-body experience, and we're trying to figure out what is the common feature in this. And I think one of the things that connects physiology to all of our molecular layers that we have, whether it's your proteins or your DNA or your RNA, I think it is this conduit of energy, right? This idea of how energy is processed and created and manipulated in stress situations. And so in this paper, we're thinking about, well, what is the energy balance? And so there's really two systems that make energy in our cells. Glycolysis, which is a tried and true path, probably the most ancient way of making energy, but it's not as efficient as mitochondria.
It's breaking down glucose and
Right. And a couple molecules of ATP that you get out of it, right? And then about 1.5 billion years ago, this bacteria hooked up with the cell, and same thing on the plant side as well. The chloroplasts hooked up with the cell, and you got these other organisms living inside cells that gave them a competitive advantage, right? Now these things, we're looking for a symbiotic environment, and the symbiosis goes both ways. What cells provided the mitochondria and the chloroplasts in animals and plants alike was an environment to safely live in. And then what the counterpart was, what those things allowed plants to do was to capture energy from light. And then our cells, it allowed us to capture energy at a very high level, right? Instead of making 2 ATP molecules, now you're making 36 ATP molecules. And so now when you have more energy, you can do more things. You can create complexity. And
let's pause there. So with glycolysis, you can make only 2 ATP.
Yeah.
And in the presence of mitochondria, you make 36. So almost 18
times more, right?
More.
Yeah.
And how many mitochondria we have in a cell?
Depends on the cell. I mean, if you look at the heart, which is where I started off, 30% of the volume of the cell, the heart, is mitochondria. I mean, the heart itself makes about 2 to 4 kilograms of ATP a day.
2 to 4 kilograms
a day.
A day.
So but your weight doesn't go up and down, right? There's no battery that stores this. So as soon as you make it, you're using it.
So that's much more than the amount of food we eat.
Yeah. It's an insane amount of mass that you're creating and dissipating.
And that's just the heart.
Just the heart.
So we're not talking about the rest of the body, and the brain also uses a lot
of energy. Yeah.
So we may be recycling our body, 10% of our body in mitochondria every day.
Yeah.
That's mind-boggling.
It's mind-boggling. I mean, the energy that's being made.
Yeah.
And that energy creates complexity, right? It allowed us to evolve, I think, as unique organisms. And the big question that's in my mind right now around this mind-body space, when we get deeper into that idea, is what part of energy creates this, right? Is it the ancient energy of glycolysis, or is it this energy that mitochondria create? And I think we're thinking of it from a unique perspective right now, and we'll see what the data shows us. But
And then when it comes to mitochondria, the other question is, how do we figure out how healthy our mitochondria are? And then another question is, what is defined as health of mitochondria?
Right.
Because is it producing ATP? Just because when I think of mitochondria and you are bringing this idea of energy center of the cell or the body, and if it is like an internal combustion engine, like a engine in our car, then the efficiency can be measured in many different ways, how many miles per gallon, or even the other way, how little carbon dioxide or toxic molecules it's producing.
Yeah.
So how do we figure out what is the definition of a healthy mitochondria?
Yep. So the best way to do this is what we've been doing, right? In our collaborative projects, you get a research level grant, which then gives you millions of dollars to do this. And you invite participants to come in, you numb their leg up, you take a piece of their muscle. And so now you have their mitochondria, their DNA, their entire environment that they're creating. In similar ways, right, if people have diseases, the best way to sample mitochondrial function is to look at the organ that's diseased and to biopsy it and look at mitochondrial functionality. Problem with that is you have to have typically fresh tissues to do it. There's groups that are working on frozen samples, but again, it's just things degrade over time, right? Fresh sample, so it limits how far away you can be from a high-end lab that can do that stuff. And then once that sample comes to the lab, it takes 6 to 8 hours to process all the different pathways and things that you're looking at. And so it's not something you can scale to the rest of the world and give people access to. And so a couple years ago, we were involved in the NASA twin study, right? There were 10 sites picked across the US. UCSD was one of them. Brenda Rana, who I overlapped with for about a week in Paul Insel's lab, we became good friends during that time. And then we interacted over the years as well, was the site PI for UCSD. And she knew we were doing a lot of mitochondrial stuff. And they were looking at some element of metabolomics and some proteomic aspects. And so they wanted to look at mitochondrial functionality. And so I went to...
Can you go back to what is the NASA twin study?
So the NASA twin study. So this was the Kelly twins, the senator from Arizona, and his identical twin, who were both astronauts. The task was that one of them would stay on ground and the other would go into space for a year. And this was to look at the impact of the space environment with all that it entails: isolation, radiation, all the zero gravity in the environment that this new body has to live in, and what the impact of that would be on the entire body, right? Humans have to adapt to everything on planet Earth. The only thing we haven't had to is radiation levels and zero gravity, right? And so what would happen to a human body in this space? The idea was that studies like this would get us closer to thinking about how to put humans on Mars and beyond, right? That we had to do this in order to figure out what the physiological thing is. One of the things that's a NASA problem, I think, and a space problem is we tend to work on engineering systems that figure out how to get things there, right? So they solve the hard body problem very quickly because you can create materials and things. But the soft body problem could persist, right? We just don't know what happens when you put a human into that vessel and all the things that it gets bombarded with. And so I think that was the focus of this twin study. And so we had samples from these twins every couple months before the one went up into flight. And then every 3 months, Elon Musk would send a rocket up and we'd get samples back. And so we had samples every 3 months during the year, and then samples a couple months after they returned as well. The idea then is, once we had the complete set, is could we invent assays to look at various elements, right? So this was probably the most expensive N of 1 study ever done, right?
So what are the samples they were bringing from space?
Blood. I can't remember if there was any microbiome sample in this at all or not, but I know there was blood samples,
Urine.
some urine, I think
Saliva?
it's saliva. And then they'd done a bunch of physiological experiments up in space as well. And so just things that were easily captured, right? And then the blood was processed in unique kinds of ways. So we had plasma, we had serum, there was whole blood for telomere assays and things like that, and immune responses and things like that. So our lab got the plasma. And so the idea was invent a way to measure mitochondrial function from the plasma. And so I've been thinking about that for quite a while, right? We don't have access to the tissues because this guy's up in space and could have gotten one, but it would rocket every other day kind of thing. And so the thought I had was plasma is a global unique capture of who you are, right? This is why when you go into a doctor and you're complaining about something that they don't know, they'll run some blood tests. And most of these are done on plasma, whole blood, and those kinds of things. But most of them have a bias, right? And it's a good bias because people have worked out the biomarkers that tag to specific things. In this, we couldn't perform a bias because we didn't know, right? There are tests that you can do where you could look at specific analytes of parts of the TCA cycle that's in the mitochondria and things that create energy, right? Pyruvate, lactate, succinate, those kinds of things you could measure. It would only be the potential of something that may or may not happen because you don't know how those things come together with the rest of that biochemistry that's there. So the thought I had was, well, we have the entire biochemistry as a consequence of every 3 months of exposure to an environment. But we also have that as a consequence of their genetics, right? Their unique DNA, RNA is making this biochemical environment sort of shape and shift. And so the idea was, could we take the entire plasma biochemistry that's created and put it on a living system that's never seen it and see how that living system now adapts their mitochondria as a readout? And so then they were interested. They didn't know which body system was impacted. They don't know what the time frame was. So we basically built a human on a plate assay. And so we bought cells that represent every organ of the body, plated them out, and then used this plasma as a adaptive transfer experiment. So now we could very quickly figure out which specific time point had the weird biochemistry that created a change in which particular cell type across this human on a plate assay. And so we were able to then develop a way to really quickly predict what that biochemistry and how it was impacting mitochondrial function. And so fast forward now, right? One of the things that scientists always look for is a way to commercialize and move their brain somewhere out into the world, and get paid for it. And so I had a company that I had been consulting with for a couple years around CBD molecules and just the cell biology of this and how to work with them in terms of cell membranes and things like that. So they were visiting the lab and they saw all the mitochondrial stuff we were doing. And the president went home and he's like, I couldn't stop thinking about mitochondria. So we had this really cool interaction. And so he formed a subdivision of the company called Discovery, Versea Discovery. And they'd asked for many, many, many months for me to leave the university and come join and be the chief scientific officer and run this company. Like, I can't quit my day job, right? And this is a recurring nightmare that I have, that I do let go of my tenure at UCSD and I go do something and then I become a technician in someone else's lab. So I will never give up my tenured position until it's time to retire. But they were very open to this. And so we were able to work out some COIs, conflicts of interest and things. And so we just started playing. And then so we were able to launch this. The problem with the NASA-based test is it's not that Elon has to send a rocket to someone's house to get the sample, but you still have to leave your house to go interact with another human being at Quest or LabCorp to get a blood draw. And most of us are averse to human interactions. And so the way we miniaturize this is we have this blood card that a lot of companies use. Comes to your home, you poke your finger, a couple drops of blood on this first window, and it wicks the serum. The cool thing about this is it solves the cold storage problem, because the minute the sample dries on this, takes about 20 minutes, it's archived, and we've shown stability out to 2 months. So you literally get a sample from anywhere in the world. We reanimate the serum, and we can really run this NASA type assay on this and give you really cool dynamic measurements of your mitochondrial functionality. And it's a surrogate, but it gets closer than any other test does to reflecting what your mitochondria are doing, right? And you can look at it from a stress perspective. We can look at it from an energetic perspective. Are you using glycolysis or mitochondria with that biochemistry? And then we've invented a few other sort of tweaked things where you can look at reactive oxygen species, and you can look at network dynamics, and mitochondria are dynamic organelles. And so you really get a complete picture of who you are from an energy perspective.
So that means let's kind of dial back to the NASA twin study kind of study where, for example, you get plasma from the twin that went to the space. You're getting that plasma and you are pouring that plasma kind of on a piece of tissue that mimics the brain cell. Mm-hmm. And you're asking what is the effect of this plasma from this twin who went to space on brain, because that astronaut's blood is circulating, going to the brain, and what it may be doing to his or her brain.
Yeah.
And the way you are measuring is how the mitochondria is producing energy, producing byproduct, toxic product, the reactive oxygen species. And you can just swap that tissue. Instead of brain, you can put a heart tissue and muscle tissue and kidney, eye, whatever you want. And then you can recreating how that person might be behaving, or the biochemically how the mitochondria is behaving in this blood milieu. So then the question is, what did you find in the NASA twin study?
So we found a couple organs, right? The one that we knew and the test predicted spot on at a particular time was muscle dysfunction. So one of the things that happens is when you're in a microgravity environment, your heart doesn't have to work, right?
Yeah.
The reason why your heart works here is because it's got to pump blood throughout your body. In microgravity, that stops. And so this is why when an astronaut lands, whether it's a couple days that they're in space or a year, there's always two people on the side of them that hold them up because they collapse, right? They can't maintain that muscle tone and functionality. And so that was easily predicted by the assay, and that was the paper in the paper that was published on this in Science. That was the assay that we put in, is the muscle specific stuff. We worked on some of the other aspects of it later, and we showed that there was
muscle mitochondria were not behaving well.
Yeah, at a specific time in flight, right? And then there is recovery that happens in that space twin after months of returning back home. The muscle does go back to normal over time, but it's not instantaneous, right? That environment does create a pressure on that tissue.
But the effect was mostly in the muscle and heart because the heart slows down.
Right.
Not on the brain or kidney or
We didn't. I mean, you see some in the brain. We didn't really see a mitochondrial toxicity in there because the brain, I think, can adapt to unique environments. And then there's a lot of countermeasures that are being done, right? They have these negative pressure suits that you basically wear these pants that your legs then act like your heart, right? They're doing the pumping because you're pushing that blood back down to the leg kind of thing. It's developed here at UCSD and Alan Hargan's lab and stuff. And so the other thing we did learn was that the immune system gets impacted. And so we saw changes in immune cells with this environment at specific times. And that correlated with some of the vaccine work they were doing with the influenza vaccine and treatments and stuff. And then the other thing that we would have predicted based on this is the endothelium gets impacted. So all of there's a flow change that happens because the blood's movement through the body is changing. And so the endothelium gets impacted in this, which is the layer that coats all your vessels, and it's going to respond to this, right? When you have forces that change, that the blood is moving around with.
So in that way, suppose that we substitute that astronaut with patient X, who has cancer or who has kidney disease, then we can actually go back and figure out, based on your assay, how that plasma or how that person's mitochondria in different parts of the body might be behaving or misbehaving, right? So without taking multiple, biopsy samples, you can recreate that. And actually, what is also fascinating for me, I was thinking about it, like we have the same mitochondria in all these organs, but the plasma was affecting from this plasma. What you learned was only certain mitochondria, like in this case, twin study, only the muscle and maybe immune cells and to some extent some other cells were affected, not the mitochondria from other parts of the brain, other parts of the body. And that's really fascinating because there is no way we can actually infer that for any patient or anyone. Right.
And I think there's certain organs in certain individuals that are adapted in a unique kind of way. And I think it has something to do with the sort of origin of mitochondria, right? You inherit all of them from your mom structurally, but then they only code for a certain number of genes. There's like 30 something genes on there. Majority of them are transfer RNAs and those sorts of things. 13 actual genes that code for specific components of the electron transport chain, this important component in the mitochondria. But there's 1,500 or so proteins in mitochondria, but the majority of those proteins come from the nucleus. And so now the nucleus is a mix of your mom and dad. And so there is this weird interplay between what's inherited from the mom and the egg, the structural component, and then how this is manipulated by things that you get from both your mom and your dad. And it creates this just complex bag of everything. And then cells are uniquely different, right? When we were first trying to develop this assay, we'd actually thought, who wants to poke their finger? No one wants to poke their finger. And so we tried doing cheek scrapes because it's the easy cell to get, and you can get anyone to spit into a tube. We could never get those cells to crack open to do the assays because they're built to be impervious to stuff, right? One of the things we did figure out along the way was how to keep these things alive for 48 hours at room temperature, which was really cool. Useless, but really cool. But that's the thing. Every cell has a unique structural feature that then protects mitochondria in unique kinds of ways, right? Mm-hmm. And they're adapted. And I think this is how organisms were complicated. And a cancer may affect your lungs, but it may do nothing to your brain. You just don't know, right?
Yeah. So now you have this product or this process where people can just drop a couple of drops of the blood on a piece of paper and then send it to a lab. And then what does the lab do and what kind of reports the people actually?
Yeah, so the lab, extracts that sample off the card and they do basically this adaptive transfer experiment, right? They move your biochemistry onto a muscle cell And muscle, we think, is a very good global capture of someone's energetic state because we have a lot of it and it consumes a lot of energy, right? Eventually, the company will come out with versions like the heart specific or the brain specific or the liver specific. But right now, as a first launched muscle specific, is just easy to do. They'll give you a report that talks about all kinds of things, right? The stress response. What happens to your baseline respiration? How is it augmented or changed in a unique kind of way with your biochemistry adapted onto the cell? It'll tell you whether you're making energy with your biochemistry using glycolysis or mitochondria. And this becomes important for, it's not a diagnostic, but as scientists, you can't help but look at the data. And we've got about 3,000 people that have run the test. And we have individuals that have specific diseases that we know, and we can see some unique pathways and things that could be predictive. And so one of the other paths that we're looking at with this company is to look at SBIR funding and other things through NIH to then develop this as a diagnostic because it could be revolutionary, right? Most people don't want to do biopsies. If you can poke a finger and get really deep insights into disease and mitochondria, could be a useful tool. And then we look at the dysfunctional elements. We can give you some assessment of reactive oxygen species under baseline and stress conditions. And then we've done this, the dynamics of mitochondria is really important. And so we've created this stable cell line that expresses a green tag only in the mitochondria. And so under a live cell scope, the entire network of mitochondria lights up. The question then becomes, well, what do you do with that? When you adopt the biochemistry, those mitochondria will start dynamic. They're always dynamically moving. And so now you can actually track this to see if your biochemistry supports a connected state, which is what you'd want, or if it causes the mitochondria to start dissipating. So if you look at every non-communicable chronic disease, mitochondria become isolated. This is how stress starts, and this is the inkling of it.
So just 1 second. You said that almost all non-communicable chronic disease mitochondria are affected.
They're affected. I mean, they become the endpoint conduit. It's energy.
So that's very profound statement. So because as we get older, we are mostly, each of us is getting one non-communicable chronic disease, whether diabetes, obesity, cancer, or dementia, and all of those diseases relate to dysfunction of mitochondria. Yeah, so that means potentially with your assay, you can also look at how, instead of, if you substitute now that muscle cell with a cancer cell, you can also ask, how is my plasma going to fuel or fight a potential tumor? Wow. So then you can also check whether my brain cells are using energy efficiently
in a unique kind of way or not. I mean, that's the gold mine in this. Is it's just, there's so many things that can be done with this. And I mean, I am a mitochondrial biologist, but during my postdoc, I became a membrane biologist as well. And so there's a caveat to that all my non-communicable diseases have mitochondria as an origin or as an endpoint. The origin, I think, is really membrane dysfunction. I think it's the membrane that starts to fall apart that then ultimately expresses as this demise of energetic systems in our cells. And I think that makes a lot of sense, right? When you create this internal and external balance, which the membrane helps to maintain, you can create longevity in this because now this system is completely adapted where that outside pressure stays on the outside. As the membrane starts to fall apart, which is what aging tends to do, is now that outside can start impinging on the inside, and then you start declining normal homeostatic pathways that keep everything in order. They start to fall apart.
So it's almost like the house and the heating and cooling system. So the mitochondria can be the water heater or the heating cooling system, and then the house is the outside wall is the membrane. So as the house ages, and if I don't take care of my house, which I do, and then the insulation falls, and
you gotta fix the windows,
or the windows, they lose the argon gas, and then the insulation goes down. So that's the membrane kind of degenerating, and that puts pressure on the heating and cooling on the mitochondria. So ultimately mitochondria, it means ultimately we first fix the heating and cooling system, then maybe if I'm smart enough, then I'll think about the windows and and cracks in the door. But
Yeah.
what you're saying is many of the diseases begin with the membrane.
That's where we think, right? And so that's one of the aha moments I had when I was in my last year of my postdoc. We had sort of created this annex in Paul Insel's lab at UCSD. Two graduate students and myself moved ourselves down at the end of the hall because we needed some extra space to isolate myocytes and do the work that we were doing. And it's a wet campus, so you can have beer and stuff. And so on Friday night, we would just kind of hang out. And my wife she did her med school through the Navy. And so she was stationed in 29 Palms during my postdoc. And so I was a geographic bachelor in San Diego. And so I had nothing to do. So I worked all the time and just hung out with these guys. And so we're drinking beer one Friday afternoon, and we would do a lot of EM in the lab, and so we would put all these pictures up that we thought were kind of cool. And as I'm drinking my beer, I look up at this picture. I'm like, oh, that's really interesting. There was this caveoli, which is what we study, these invaginated caves that George Palade had discovered in the 1950s, and it turns out there's a huge biology around this, that not all membranes are created equal, that these are lipid-rich regions of the plasma membrane. They contain a lot of molecules and a lot of signaling happens in this tight space of the membrane. So there's these series of paper or pictures that I noticed on the wall where this invaginated structure had this mitochondria just below its surface, right? And so the thought was, is this just by chance, or is there a physiological importance of this interaction? So we started looking through the literature, and it's in every cell type, but no one's ever commented on this. And so now we started studying this domain interaction in the heart, and it turns out it makes perfect sense, right? The place where you would have energetics, we think this is how oxygen gets into the cell. We think that this is where all the signaling happens. This is where you get exchange of membrane and mitochondrial components. There's tons of things in mitochondria that don't have a targeting sequence that takes them to the mitochondria. This physical relationship would make sense, right? And so this was what my first R1 that I got from the NIH was based on. And it was an EM picture, pictures worth $1 million or whatever it was at that time. And it was this unique picture where we showed the membrane interacting with mitochondria and forming this dense contact to create this transfer system. And so I think there is this intimate relationship with stress and the membrane and energetics, and it's this tie that brings everything together.
Yeah, so based on this caveolin, which I want to again go back to some analogy. So it can be the doors and back door or doors in our house. And usually next to the door, we have from outside, maybe calling bell or camera or whatever it is.
Yeah.
And then inside there is a lock and key. And then you have to make the doors lighted. So there is light and all that stuff. So there is power involved to make it. And if you have a remote control door opener or the garage door opener, then there's also power involved in exchange of information. So that means the cell membrane, when you look at this cell drawn, this round thing that we typically draw, which has no definition, it doesn't actually say where is the door, where is the window to the cell. There are actually specific part of the cell where they are kind of designed to be this information exchange port. So those are the caveolin.
Yep.
And so that's where also cholesterol is
heavy, highly enriched in cholesterol.
Highly enriched in cholesterol, because cholesterol brings fluidity to that part of the.
Some rigidity, right?
Rigidity,
but
yes.
Cholesterol also binds oxygen, right? So there may be some biochemistry that brings as well.
So how is that research going, or where is that going?
We spent 20 years beating a dead horse over and over again. Lots of NIH grants, and I learned a couple lessons along this. Right, I thought the point of a scientist was just to be altruistic and publish all the things you discover. Turns out that's the stupidest thing you can do, because then no one cares. And so we have the cure for heart disease that we published in a series of papers, and now you can't get a patent around any of that because it's all in the public domain. And so we learned our lesson because we were like, oh, we should have patented all of that. And I get a lot of when I would give these talks, we had a series of papers in circulation, Jack, and some that showed that if you upregulate caveolin in a cardiac cell, you actually preserve its ability to adapt to heart attacks. You can the cell is protected from hypertrophying, so it doesn't grow big when it is in a heart failure model. You can make the animals diabetic and they get morbidly obese, but the heart stays pristine. And you can age these animals to 2 years of age and they function like they're 3 months old. They don't age from a heart perspective at all.
Wow.
And it's all you've done is made twofold extra protein in the membrane for this caveolin. But all the heart stuff was lost. And so then we learned our lesson and we patented everything on the neuro side. And when we first started this, Brian Head, Brian was a graduate student when I was a postdoc in Paul's lab. He was the first postdoc I recruited when I left and started my own lab with this collaborative group of individuals. And so Brian and I were both working in the heart. So I said, well, we can't do that. So I kept the heart stuff and I said, you work on the brain, right? You launch this whole thing. But it was a large initiative. All of us worked on the thing. And so Brian, myself, and then the two MDs that were working on this project, we co-invented and patented all of this stuff on the brain. Happy to report that it's now moved to a stage where they're going to be putting this into humans in probably the next 6 months for ALS, right? It turns out that neurodegeneration, this protein degrades. There's a family in the UK where you have an actual mutation in caveolin gene that tracks with ALS. And so now a company has picked up on this patent and they're moving very quickly towards IND and putting this gene therapy into humans for ALS. Brian just had an amazing paper published. So this goes back to this idea of why don't drugs make it to market? Because they don't use the right models. And so most models, if they're doing young animals, it's never going to work. But then the other thing that scientists do, because they want to bake in the best effect, is they do it as a pre-treatment. Right? So absence of disease, they give the therapy and then they create the disease and they're like, oh, look, it's protective against this disease. If you could predict when you have Alzheimer's, great, but most of us can't predict this, right? Yeah. And so he did just an amazing experiment where it's entrenched Alzheimer's in these animals. So they're full-blown Alzheimer's model. And then he did the gene therapy and it reverses the Alzheimer's, right? Truly proof of concept and principle for this. And so we think ALS is the first target, but this I think is going to be a gold mine for Parkinson's, Alzheimer's, anything neurodegenerative related, caveolin's going to fix because it brings back all of the signaling by just putting very little energy in the system. All you have to do is express one protein.
Yeah. So this is like the scaffold that brings everything together.
Yeah.
And then they can signal, mitochondria will supply energy and back to business.
So I feel like my circle is done with this, right? So we brought this, we're still doing some side grants on diabetes and other things around caveolin. But I think the lab is really fixated on this new area, this mind-body connection thing. And I think, I mean, this is the thing. There's serendipity in things you always have. But I think in some of the things that we're doing in the mind-body research, I think it's going to come back to the membrane, right?
Yeah.
I really think that's how the physical and the non-physical world and all these things interact is at this level of this membrane.
But also another point that you made was when scientists, we publish certain things, we don't think about the global implication or the patent. And,
right.
And then what happens is there are a lot of knowledge that are outside in the public domain that could actually be leveraged to develop new therapies, new treatments. These are powerful, but then since those are not, nobody filed a patent, so then
no one can make money on it, right?
Yeah. So I also remember when we first discovered melanopsin, I was with GNF, and then they said, well, nobody will make any money out of melanopsin because it's a blue light receptor. You cannot patent the sunlight. But then when now I look at it, there are a lot of people who are coming up with stupid blue light blocking glasses or blue light emitting diode and all that stuff. But nothing actually has even a criteria how much blue light they should be blocking or how much blue light they should be emitting. And then in the absence of patent, almost every Tom, Dick, and Harry is producing one device.
Yep.
That's that's kind of contaminating the
And they make outlandish claims about this.
yeah, they make outlandish claim about it. So sometimes, you know, there may be public domain knowledge that's actually extremely powerful, but since it's not IP'd, that's
Yeah.
not leveraged. And conversely,
Yeah.
there are also some really half-baked ideas, since they were IP'd, both government and the pharmaceutical companies also spend a ton of money
Yep.
to bring them to.
Yeah.
It's like they beat the dead horse for very long time.
For sure. And there's so much pharmacology out there that's just based on that kind of thing, either
Yeah.
bearing stuff as competitors or bringing to market drugs that are, it's too big to fail at a certain stage, right? You spend so much money. It may not be the best drug, but we've spent all this money. We got to make some money back, right? It's just sad.
Okay, so now we are back to, you said that all this discovery on caveolin kind of brought everything back in full circle because you connected the brain health
Yeah.
with heart health. You started with heart health and ended with brain health. So that brings up your other life.
Yeah.
That is the meditation part.
Yeah.
And of course, we started with the idea of philosophy and how it influenced you. And meditation is always linked to some aspect of philosophy or faith.
Yeah.
There are two parts question, and you can prefer to take which part first. First is how did you get into this research on meditation? And then the other question is, when you think of meditation, it always goes back to the thought of faith. And how does meditation link to faith, or should faith be part of our overall health?
Yeah.
So these are two different parts, and so you've got plenty of time and pick which one you start.
Yeah, so I'll do the easy one, which is how do I start on this? So I've always been, I grew up as a Hindu. I never really meditated, but when I started exploring that aspect of my background in college, it was two Germans that shaped my theology. One was a German who went to India and became a Hindu, and so he taught all the Eastern religion classes at the university. And the other German was the local Lutheran pastor. And so he was just an amazing guy. Mark Appold, he was a theologian. So he got his theology degree from Germany. And then was in this small little town, and he was the pastor at the church. And so his outlet was, he taught at the university. And the smartest person I've ever met, right? He spoke 7 languages, all of the ancient ones, so biblical Greek, Aramaic, Hebrew, English, German, just a Renaissance guy. But he could carry a full-on discussion about genetics and give you a 2-hour sermon on the Bible in the same breath, right? Just one of these magical individuals. And so he really shaped my Western theology. I took every class that he offered on Jesus and Paul and the Bible and all, and I actually even took biblical Greek. And so for a certain time, and I was into ancient languages as I mentioned, a nerdy kid, right? So I took Latin in high school for 4 years, took 2 more years of Latin in college, and then took 2 years of biblical Greek as well. Useless languages. I can maybe pick up a Greek book and make some of the letters out.
I think our next should be Sanskrit.
I know. I've heard it's one of the hardest languages to learn. But just this idea of doing that. And so meditation was a part of that experience. And so from Lloyd, the Eastern theology teacher, he taught me how to breathe. And so he was my guru. And so I took this Western or Eastern theology class from him. And as part of that, he would have you out to his place and he would teach you how to do mantra meditations and things, and then to do aspects of yoga breathing. And so he taught me how to do like the Ujjayi breath. And then he and I share this cosmic bond because he, looks at what you want and he whispers this mantra into your ear. The only time it's ever said is when he whispers it in your ear. And you're forbidden to ever say it out loud again. And he and I are the only ones that know what this is.
But you can say it loud when you're alone.
Not verbally out loud. You can say it in your mind, but that's the other thing is, right? You're not supposed to conjure enough of it around in your mind. And this got me through very little sleep in college. These things. It's amazing what your mind can do when you give it a mantra and some kind of focus and energy from that perspective. And so the way I got into, and then life happens and all of that falls apart. And so we left college and, grad school and all these things happen. And so for about 10 years at the VA hospital, so I moved up after my postdoc. I had a junior faculty appointment at the VA in the Department of Anesthesia. So I moved from pharmacology to anesthesia, which is a niche pharmacology department essentially. And the social experiment that the department was doing at that time was into an same office an MD and a PhD and see what happens. And so I got paired with this pain anesthesiologist, Toby Muller-Bertram. And Toby's just this larger than life German who's just very smart and very just focused on things, right? And so Toby and I would talk about lots of stuff. And the idea was that we would start a project together. Never did any science, became really good friends. He left the university. We stayed in this office for 10 years together. So we got to know each other really well. He left the university and started a pain service in the Palm Desert area. When the opioid crisis hit, he started experimenting with alternative ways to manage pain. And so one of the practitioners in his clinic was this chiropractor who practiced a lot of meditation with this guy, Joe Dispenza. And she brought some of these meditations to the clinic and they saw crazy effects on pain. And so Toby went to one of these events and was just blown away, right? I mean, you came to one a couple of years ago, right. And unless you experience it, there's no way to describe it. It's a magical journey into a transformation of self, right? Whatever you think you can do, forget it until you go to one of these 10-day, he's doing 10-day events now, 7-day events. And so then Toby remembered all the crazy things I think up and do. And so when he wanted to start working in this space, he came to me and said, hey, we should work on this project together. And so that's how I got involved in meditation. So I had that background in philosophy, religion. I'd done meditation myself. And so now this was a way to think about it from a biological perspective, right? What was happening? And for me, and serendipitously at the same time, I was involved as the vice chair of research of the department in chairing a committee that was recruiting a new faculty member. And the one that we had ultimately settled on was Fadel Zaiden, who's this world's expert in mindfulness meditation. And I was looking at his CV and sort of learning about that space. This was a hot area in anesthesia, right? How does mind control body from the perspective of pain? And that was what Fadel's focus was on. One of the things I noticed in this, and I'm not a neuroscientist, but I can pick up a book and read about neuroscience and figure things out, was that most mind-body researchers tend to fixate on mind, right? So there's this hyperoptic focus on, well, what's happening to the brain when you're meditating? Seems like a no-brainer, literally, right? The brain's going to change. So who cares? Let's move on. Let's move to the next phase of this. Let's see what the brain is doing to the rest of the body, because there's a different thing attached to it. And that's what became our niche focus, and I think really differentiates us from everyone else. We're looking at the brain in sort of rudimentary ways that most people do, which is, EEGs in a large scale, and then in really refined ways, fMRI kinds of things. But the bigger question is when that brain changes, in whatever way it does, and we know it changes in unique kinds of things, what does it do to the rest of this, right? And what does it do to the rest of this when it's healthy? And what does it do to the rest of this when it's not healthy? And could we now start thinking of this arc of brain-body connection, and could this reveal some things about our condition and how we can heal, right? And this goes back to the stuff I was doing as a grad student. We had discovered these latent systems that exist in our bodies that when you coax them by stress, holy crap, the heart doesn't get injured. And it's the same thing with this mind-body connection. I think these are ancient things that have always existed within us, and we've just forgotten to use them, right, in the way that we should. And I think this was an opportunity to uncover some of that. So that's where we really went into this aspect. The faith question.
So let's hold on to that. So this meditation, and you started answering with going back to your college and talking about breathing, control of breathing.
Yeah.
And then when I think of meditation, actually, I grew up slightly differently in the sense I grew up in a my both parents were very religious in the sense that actually I had to get up early in the morning and then also had to do some little bit of meditation as a child, like I was 10, 12 years old. So I didn't know too much. But then my father essentially told me three things that I should be doing when I'm meditating. First thing is control your movement. As a child, you're always fidgety, so you have to sit still.
Yeah.
And that's a lot of control, self-control you have to do to just sit still.
Yeah.
And in a specific posture, like your back straight and all that stuff. And I think that's where you engage your nervous system, your fine motor skill to make sure that you're actually constrained. So it's the opposite of control movement as in dance or sports. And then the second thing is learn to breathe.
Yeah.
And breathing is the only, autonomic system that we have some control over. We can stop breathing for a while. We can regularize breathing. So I think this is only one thing that we can actually do. Another thing, except for controlling our movement. And then the third one was, just like you said, mantra meditation. So that means you are actually focused. The mantra helps you focus your mind from not wandering. It's just, again, controlling, just like you're controlling your body not to move around.
And your mind as well,
your mind not moving around. So it's a lot of control that you are exerting on yourself.
Yeah.
And it begins from the brain, because the brain is always making my hands and legs move. It's also having a default mode network that's controlling my breathing and other stuff. And so I guess in one sense, meditation is some kind of exercise for the brain. Then another point that we kind of discussed almost synonymously, but actually they may not be synonymous, is the mind and brain. What is brain and what is mind? And then if we go one step up, maybe consciousness.
Yeah.
And how do these three come in? And I guess that takes you, I think that's where we can also segue to the second question, how the origin of meditation in different cultures. And actually meditation is almost always, except for the Chinese culture, I think, it's always linked to some kind of religion.
Yeah. I mean, I think for me, that arc has been kind of interesting. So, meditation is part of Hindu culture, right? I mean, it's whether it is or not. If you go to India, most people don't meditate, right? If you ask them, "Hey, do you meditate?" Most of them don't. But it's ingrained in the ancient culture, right? It comes in all of the texts and stuff. And you see it, right? There are yogics and other things, and Brahmins and other ones that really pray and meditate and do these kinds of things. But every culture has some element of that. So the other thing that I went through in this arc of my development was that even though I was raised a Hindu, I've never really sort of believed in this concept of God, right? I didn't know what that was. It was an abstract thing. I knew something bigger and better than me existed. I didn't know what that was. And so I called it spirituality, right? It wasn't a particular person or an energy or an idea. I just knew I wasn't it. There's something else. And I think the grounding force for all of that in my life was my mom. And then she passed away suddenly at the age of 60. And it's now been, 11 years. And it rocks everything you think about. And so this is me in my 30s. I've been married for a number of years now. We have 3 kids. And one of the things I learned in Western theology is that the kids always get raised with the faith of their mom, because that's how things track. And so all 3 of our kids were baptized and they grew up in the Catholic Church. And I would go to church every week because it was part of I didn't believe, but I'd read the Bible and I knew that Jesus was a historical figure and all these things, but that faith thing was always elusive. And so after my mom died, I went into this, sort of a depressed state. And I had these series of dreams that led me to this conversion. It was just a weird, bizarre set of dreams. And the ultimate resolution was I had spent all these years, all these weeks sitting in church, and the Catholic Church is very graphic in the sense that they have this crucified Christ sitting on a wall. And my sort of question was, well, if I've spent all this time and energy, why don't you come off that cross and talk to me and convert me, right? And I was watching this Netflix series on the Bible, and I like shows like that that are historical in context. And there's this one scene where it's the risen Christ. So Christ dies through a series of events that happen, and it's sort of this passion of the Christ, and he suffers while he's dying. And then the magic of Christianity, or the belief and everything that comes out, and I say magic because I don't know how to describe it or explain it, is that he rises 3 days later, and it's him conquering death. And that's what the faith is built around, that you have this immortality based on what this God figure did. But then there's these apostles, his closest confidants that he was trolling the countryside with. And one of them is lamenting the fact that everyone has been able to see the risen Christ except for him. And Christ is sitting in the doorway, and this apostle can't see him. The minute he breaks the bread, which is what Christ teaches you at the Last Supper, this how will you remember him through this exercise, right? And early churches were thought of as cannibalistic because they were eating the body and blood of Christ, but it's this imagery that's tied to this. And there is, in Catholicism, it's really truly the blood and body that becomes transformed during prayer and other things. And so when they break this bread, Christ becomes instantly visible. And so for me, that was the moment where I realized I hadn't done what I was supposed to do. I'd sort of been there, but I hadn't gone through that conversion where I could partake in that relationship. And so the Catholics don't make it easy. So it was a year-long process.
Hindus make it impossible.
I would think so, but it's
You cannot convert to Hinduism, right? So they make it impossible.
And both the Hinduism and Catholicism have this general feature to it, right? That they're all accepting and all encompassing kind of thing. And so I went through the Rite of Christian Initiation for Adults, this RCI program, which was a year-long program. The first class we were in, it was like 7 or 8 people in this class. And you get to know these people really well because they're going through this process with you. The first lecture was on this concept of faith, hope, and love. And I'm like, all right, I'm ready because this is what I struggle with all the time. And it's this guy, Bishop Barron, who runs an interesting podcast on Words on Fire and kind of thing. And so he describes faith in a context that a scientist can completely buy into this, right? I'm like, I'm hooked. This was amazing. So what he calls faith is not this sort of Kool-Aid drinking thing that most people blindly believe in, but that it's really a very calculated belief in something, right? That there's this circle of reason, and faith is just outside that circle of reason, that you just haven't asked the right questions. You haven't created that data set that gives you the observational things to move from that reason circle to that faith. But eventually, we as scientists do this all the time. There's things my brain comes up with that there's no proof for, and it's faith. I have to use that faith to convince a postdoc to do that right experiment. So now that circle of reason, once we do the right experiment, gets bigger. And now that thing that was in faith becomes a part of reason. And so that appealed to me, this notion that I just don't have enough experimental evidence to show that Christ really existed, and that I know he existed as a person, but that he was the Son of God. Who's to say he wasn't, right? And that's that belief, faith aspect. And so it's the leap you take. And so that's the leap that I sort of jumped into during this year, is that I'm going to believe, because I have no proof that it didn't happen. So I have to contend that maybe that eventually I'll gather enough data. And so fast forward to Easter, which is when you become a full-fledged Catholic then. And so there's 2 things. It happens really quickly when you're an adult, right? As a kid, it takes years to go through this process. So you get baptized, you have your first communion, and you get confirmed in one evening. Whereas as a kid, this happens, you're baptized when you're young, you get your first communion like around, a couple years when you're older, like, couple like 3rd or 4th grade. And then you get confirmed when you're a high school kid. So all of this happens in one day. I thought the magic was going to be the baptism part, right, where you have a full immersion. And there's an interesting story that ties back to my mom. As an Indian kid, everyone has a star chart that's read, right?
Yes.
That dictates what the syllables of your name and everything about you. And so my star chart said I was going to die in water. And so I was not allowed to go on boats. I didn't learn how to swim, which was really an impediment in white America because everyone swims. And so I had this aversion to water my entire life. And my wife pointed out, do you realize you're going to die in water today? And so that's what a baptism is.
Yeah.
You essentially get dunked Father, Son, Holy Spirit, and you die and you become a new person, right? And so that was this Indian Hindu star chart was realized in a Catholic ceremony, which is just crazy how the world and the universe is. And so then nothing really happened because it was traumatic for like water. It didn't change my perspective. I didn't have this connection to God. And so then you quickly change and then you get ready for your first communion. And so the communion is you are going to ingest God, right? So the priest prays over this bread and wine. And every religion has this as well, right? In Hinduism, it's usually a copper vessel and it's water until the priest does his whisper thing over this water and it transforms it into a holy conduit, right? So the priest prays over this bread and wine, and it becomes the body and blood of God, Jesus Christ. And then you ingest this. And so I went through this process, and it was my first time. And I can drink decent amount of alcohol and be totally fine. I had a tiny sip of this wine, and I lost my footing. I could not stand up. That was my first mystical experience I think I've ever had, right? This connection with something that was completely outside of me that really was born out of this faith. And so then the question becomes, do you have to have this faith background to touch this, whatever it is, right? Whether it's God or Jesus or light or energy or power or some spiritual being kind of thing. And I've seen this at these events. I think faith helps because it gives you this ability to go beyond this edge of reason. But I think an atheist can get there as well. It just takes a little bit longer, possibly. But it's doable. And I think it's the constructs that we build in our heads to connect us outside of this. And I think what you mentioned is one of these things, this notion of what the brain is, what the mind is, and what this higher level consciousness is. I think we're all able to understand this. But I think there's a humanness to it that connects us in a unique way. One of the studies that we're onto right now that we're trying to do is there a genetic basis of a mystical experience, which would then tie to this consciousness state? There's a pharmacology to this, right? And I think the easiest access is the plant medicine. So there are certain individuals that will take a tiny drop of psilocybin or ayahuasca or something, and they will have a full-blown mystical experience from this. And then there's certain individuals, you give them a gallon of this stuff and they won't respond. There is not a linear relationship. This to me suggests there has to be some innate genetics that this pharmacology is interacting with that allows some people to achieve and others to not. And so I think you can self-induce this through meditation and breathing and other things. And I think there's a unique genotype that this interacts with. And I think that's one of the things we want to identify is, what is this? What is the consciousness aspect of it? And how does brain, mind, heart, how do all these things connect and interrelate? We'll see.
Yeah. So coming back to this meditation, your interaction with Toby,
Yeah.
going back to going back to that topic. By that time, Toby was already in Palm Spring, and you were in VA.
Yeah.
How did you again connect?
So Toby is this eclectic individual. So he collects houses. So he had an apartment in Palm Springs that he would hang out in, but he kept his house in South Park. So he was always in San Diego back and forth. And the one person that connected the two of us, so he grew up with this guy, Jan Schilling. I don't know, I can't remember if you've ever met Jan.
No.
We may have done some way back ways work. Mm-hmm. So Jan went to medical school in Germany, and When Toby came to the US, he wanted to help Jan transition to the US. And so Jan came and worked for Toby in the clinical setting because he wanted to become a physician here, and this was a way to get into the system. And then there was a slight delay. And so Jan then wanted to work in research. And so the slight delay turned out to be over 10 years. So I worked in my lab for almost 10 years. And he was a German, right? These Germans are amazing. Jan was just, he ran the lab. And he was one of these guys where if there was a problem, he's like, I'll figure it out. And it's just a guy you could build a lab around. And so he did all the mitochondrial stuff. He did all the learning, memory, behavior kinds of things that we developed in the lab. And so he became the conduit for this. And so he's the one that connected me and Toby throughout this whole time. And so Toby was always around hanging out with Jan, and so I would join them for coffee and stuff. And so we continued to interact.
So you are influenced by quite a few Germans.
A lot of Germans, yeah.
Is your wife's ancestry German?
She has some German, Norwegian kinds of roots, right? European. So maybe there's this connection. I don't know.
Yeah. So when did this meditation, when did you start going to Joe Dispenza's meditation camps, and what was your first experiment there? And
the first one was February of 2020.
Oh, February 2020.
And then you know what happened in March.
This is the BC. This is BC, before COVID.
Yes. And so we had designed a study. We had a donor come in, and she came to campus. We took her to the faculty club, and that night I hadn't known, but she had written a check for $100,000 to get started, which I could spend in 2 days, probably less than 2 days. And so we planned our first study. It was to look at 30 individuals, all healthy, and see what happens to them from a brain, heart, and sort of biological perspective. And so there was a German team that Joe was using to do EEG measurements. And then we went in and drew blood, right? This is what Toby does in the clinical setting and was really good at it. And so we were able to create a data set around this. And so when the pandemic hit, these samples just sort of sat around for a while. And then we started exploring. And then we actually built a COVID story around this. And it turned out that there were factors that were being upregulated by meditators that could actually prevent COVID infectivity. And we described this in a paper, and just really cool biochemical studies that we did around this.
So just wait a second.
Yeah.
So by meditating, these people could actually improve resilience against COVID.
Yeah.
And what was the study? So you'd go to this. Can you describe a typical Joe Dispenza?
Yeah, so it's a week-long event, right? And so this first one was in Indian Wells. I spent about I spent one night there and a couple hours in the thing. I was not expecting much to come out of it, right? It was basically, it seemed like a rock concert because I was expecting like some mystical environment where there's lavender everywhere and just calm and collected. And they're dancing around and moving. But then when they were meditating, right? They're creating a space and an
How
energy.
long do they meditate for?
So it's 7 days, equates to about 35 hours of active meditation. So 17 unique meditations they do during the week. Sitting, standing, walking, all kinds of different ways of doing it.
5 hours of meditation every day?
Roughly. Yeah, roughly about that. And then there's what we've academically been calling reconceptualization, right? So there's teachings that go with this as well. I think this is the big problem in a lot of meditation styles, is that you're thrown to the wolves, right? At least your dad gave you 3 targets, but most people like go in the corner, close your eyes and achieve, right? And most people can't achieve because they don't have a target. They don't know how to get there. And what I think Joe does masterfully is he teaches people how to practice and fail and practice and fail. And he gives you that room to do that as an individual, right? And there's lots of abilities to do this. It's 17 chances you get at an event. And then when you go back, you can continue on. And so it's an intense retreat. My first one that I went to was in 2021, and it was in Orlando. So I'm like, maybe I'll get to go to Disney World. No time, right? About 55 hours of commitment during that week at the event. So it's really an immersive event. Mm-hmm. And then as a scientist, it is the perfect petri dish of an environment, right? Because everyone's on the same sleep-wake cycle because you're expected to be in the room, and people pay to be there. And so 99% of the people are there. I've given lots of talks in ballrooms and stuff. I've never been in a room where no one's looking at their phone, writing stuff down. I mean, they're writing actual notes and things down, but they are intently focused on capturing the information and applying it in a unique kind of way. So it's a unique environment to be in, right? And it has that element. Food is at a resort, so you're eating similar types of food. You're not changing your diet. So if you're vegan, you're vegan. If you're gluten free, you're gluten free. If you're carnivore, you're carnivore. But your timings are exactly the same as everyone else because you're guided towards that. So it becomes this amazing environment to then study the impact of meditation on a very large group of individuals.
And there are like what, 1,000, 5,000?
The biggest one I've been to is 2,500.
Yeah.
Most of them are anywhere from 17 to 2,200. And so you have, and these are people that want to be studied, right? I've never had a problem recruiting for a study. And this, one of the problems was recruiting for studies, right? The first ones we recruited for, we were doing controls. And so they were such involved studies that we could only have 30, 40 people. And we would get 700-800 volunteers for these 30. So we would have to randomly select 30, 40. I go to the event because we're there running the labs and stuff. I get so many people come up to me and say, what do I need to do to be in your study? I've tried 3 times now and I've been rejected all 3 times, which then led us to think of this idea of, could we invent a study? And this is where these quantum level studies came, quest to analyze 1,000 humans meditating. So the idea was to recruit as many people that wanted to participate. The only exclusion criteria is you refuse to wear a wearable device, right? If you didn't want to do that, couldn't be in the study. And we've run now 3 or 4 of these, and it just collects mega amounts of data.
Yeah, so that's very exciting because, I do another type of clinical trial where we have to go through maybe hundreds and sometimes thousands of people to get even one participant into the study.
Yeah.
And it takes, in a typical month, in a happy month, we get 4 to 5 participants into our study.
Wow.
And I envy you tremendously because you within 15 minutes,
Yeah.
you get all your participants.
And in 7 days, we do a study on 1,000 people.
Yeah. So you bring these people, or these people come there. You can take a blood sample or any sample from the beginning of the study when they haven't started meditating. And as you said, there are a lot of people who have never meditated, that intensely. To be frank, when I went, I had meditated to some extent, but I never sat still for 3 hours.
Right. That's a long time.
It's a long time.
I can't remember. Did you do the pineal? Did
No,
you do the long? Okay.
I didn't stay till the pineal.
Fine. Yeah.
But it's as you said, there is music or the background music that helps you breathe, controlled breathing.
There are, there's unique repetitive beats in there, right?
Yeah.
And so one of the things that I learned early on, someone was helping me get through my first event, and she said, focus on the repetitive sounds you hear in the music because it will calm your brain. And I started doing that. I'm like, holy crap, this really works, right? And it's patterns on patterns on patterns, and it's in the music. And if you fixate on it, it's a good way to focus your mind.
Yeah, actually, that reminds me. So long time ago, Ravi Shankar used to live in San Diego, and he used to, I have another family friend who used to organize these home concerts. Mm-hmm. So that's where, Ravi Shankar was his neighbor. So there are many musicians who would come visit Ravi Shankar, and on Saturday, Sunday, I think it was on Sunday afternoon, he would invite some visiting musician. They would perform in this living room, big living room, without any sound amplification. So there was no amplifier, no microphone. And as a student, it was awesome for me because I would go there for $10 and then listen to the music. Sometimes Ravi Shankar, sometimes George Harrison, or famous musicians from India, Zakir Hussain. And there was one instance I clearly remember because Ravi Shankar was telling us, explaining what are the beats and what they do, how they repeat, and then he started playing. And I closed my eyes and I could feel that mystical experience, maybe towards the end of the concert. It used to go for a couple minutes. When Ravi Shankar used to play, he would go for 1 hour. So that reminds me that this music and the beats, just like they help us exercise. When people run, they have a specific
Rhythm,
preference,
right?
rhythm. So this is also one thing that we forget, that meditation, people always think of meditation as sitting in a calm, quiet room and trying to control.
And mantras are an element of this, right? So the one that I share with my guru, right? It's never said out loud, but it's resonating in my head as I'm meditating. And it's what allows you to focus. And so one of the things we worked on a lot was my struggle, and I still do this when I try to do it at Joe events and stuff, is my brain is so active that thoughts continually come in and they disrupt me going into a weird space. And I think a lot of us struggle against this and we try to fight it. And one of the things I've learned through, religion and other things is that that's the wrong way to let those thoughts come in. Just let them come in and let them move through and flow, right? Eventually you will get to a state where those thoughts stop flowing, right? And you're now in this perfect, peaceful state. And I think this is what mantra and music and sound allow you to do, is to take in all of those things. And I think the other thing I think about is the reason you have those thoughts is that you need to work through something, right? And until you've worked through it, your mind is not in a place where it can touch and feel that mystical state. And so I just sort of let it happen now, I think.
Yeah. So in these experiments, so the first one was COVID, so that was 2021. And then ever since, what are the different experiments or questions you have asked? Because, I have been to one of the events and I was really, I was super excited, means there are so many ideas came, just being there for 2 or 3 days. So you must have had tons of ideas. So what are the key experiments that have gone through the process? Because it takes, any good idea, it takes 4 to 5 years to go from inception to publication.
So there isn't a lot in the literature around. So I guess we'll start at what's in the literature, right? So I think what exists is that there are certain omic domains that people have looked at. The one that almost 90% of the studies look at in this space is transcriptomics. Cheap, easy to do. You can do it in isolated RNA, that kind of stuff. Fewer studies on metabolomics, proteomics, almost no studies, maybe a handful on microbiome kinds of things. And so what we wanted to do was apply a multi-omics set to this, right? This idea of what's happening across all the layers of biology, and are these integrated in a unique kind of way? The first big quantum study started with microbiome because no one had really looked in large scale. And there's also happy serendipities that happen, right? Originally, we'd focused completely on healthy individuals, which I think is important. I think one of the barriers that people have in this is they think that there's a toxic effect of going to an event like this. And we showed that a healthy individual comes out of the event healthy. Nothing bad happens to you kind of thing, which is important. But then during the quantum study, in our first quantum study, we recruited 970 plus people, right? So it was almost 1,000. So we got our quest to analyze 1,000 humans meditating. What it turned out when we started looking at the demographics is there were over 50 diseases represented in that 1,000 people. And so now not only were we asking the question simply of what happens to a healthy individual as they go through this arc, but as a pharmacologist, I'm thinking of how a drug company designs a trial. They'll spend $1 billion to cook up one drug for one disease. Now I've got one drug, which is Joe Dispenza week-long event, and I can look at how it impacts 50 diseases at one time. And this is the arc of what we've been doing, is all the quantum studies are built around this notion of how does the individual change when they go through this 7 days of meditation? And is there an impact on disease in this unique way? This starts to ask that deeper question of mind-body from a very materialistic component, right? How does the focused mind change the materialistic body from a biological perspective in the setting of all of these diseases, from the level of cancer, which we can pinpoint to a specific organ, to diseases of the mind, which is probably the largest. About 45% of the individuals in our database have a mental health issue that they're working with: anxiety, depression, PTSD, those kinds of things. And so now we can ask those unique questions about material diseases versus manifested diseases like the mind, which may have a material component to them as well. The other thing that we've started to look at, and I think one of the things that this sort of begs the question of, is the individual goes through this event, but me sitting there observing and watching all this, it turns out that that's missing the scope of everything that's happening. We are a socially connected organism. We tend to thrive when we're connected to family and friends, and the blue zones have lots of issues, right? There's lots of things that have come out that suggest that people don't keep accurate records and things like that. But I think one thing that does survive with the blue zone is connectivity. I think that it's not herbal tea or special water or exercise, or it's who you connect with, right? You can smoke and drink, but as long as you're doing it with a friend, it doesn't have the negative impact that it typically does. So social interactions matter. What we started to do now with the quantum studies is look at the larger dynamic social interactions that happen at these events. When you have 1,000 Garmins in the room, you can ask and answer this question. You can see what the Garmins are doing when everyone's not in the room and they're in their separate spaces. And you can start to look at now what the Garmins are doing when they're all in the same room doing the exact same thing. And we have a very simple thing that we've looked at with the last quantum was about 750 people. We had data from 700 individuals 24/7 from the Garmin, like usable data, 90% accurate. What we see is there's about 250 people that are healthy, and we have 50 in that initial cohort that are depressed. And when you overlay the entire signature for the full event and you codify when they're meditating together and when they're in and out of that space separately, what we see during those separated times when they're not together, when they're listening to the lecture, when they're not coherently interacting in a space, creating something, is that there's separation. What we see are there's unique differences in these patterns of HRV, where the healthy have a very smooth curve throughout, whereas the depressed individuals have a very jagged kind of thing. So they're struggling in some way, and it shows in their physiology. When you look at those same individuals during the meditations, there is this eerie alignment of their HRVs. They look like they're twins, the 250 healthies and these 50 depressed individuals. And by the end of the week, what we see is that in the spaces when they're not together, they start to look more aligned, right? And so there is this amazing thing that socialization does at scale where it brings us together, but it changes our physiology. And so this is the larger question we're asking now is it just the individual's problem going through and creating this consciousness state, or can you accelerate this by creating a group dynamic, right? The group becomes important. There is this living, breathing organism that's the larger human sort of connection. And I think that's what these types of events create. And we're starting to look at this, and we're going to do this on steroids in Denver, which I wish you could come to this, but the breath thing becomes really important. And so I have and this is this crazy connected arch. So the student that trained in Garrett's lab for his PhD after me was Eric Gross, and Eric was an MD PhD. We tried to recruit him to UCSD as a resident when he was sort of making his decision. He ended up at Stanford for some stupid reason. I don't know why, but anyway.
Yeah. Why people go to Stanford?
No, I don't know. But he's done really well at Stanford. Eric is a really smart guy. But he was working with Daria Mockley-Rosen, who was doing a lot of the PKC stuff and the staple, the peptides and things. And so with her, they also had hit on this idea of these aldehyde dehydrogenase mutations in Asian populations, the flushing gene. And so he does these experiments where people drink really high-end vodka and then you breathe into his really fancy breathalyzer and it looks at aldehyde decay. And so the idea, we meet up once a year at a pharmacology conference. And so we were talking about this. I'm like, this would be really interesting to look at in meditation because a lot of it is breathing. And so the idea is a meditator creating a new breath at the end of the week, and breath becomes deceptive of diseases we possibly have, right? Lung cancer and other things. You're creating other organic volatiles that you're spitting out into the things. So we did a pilot study, and it was some interesting things we saw. We basically have people breathe into bags. We'd ship the bags to Stanford. He'd run through his mass spec again. And then we tried to get a mass spec on site. It didn't work out. But there's a company in Boulder that has the fancy mass spec, time of flight mass spec. And one of the people that we're interacting with, he has a company that does mass spec stuff. And so he knows these guys. So he facilitated this machine on site. And so in a few weeks, we're going to have this system sitting in the room. And we're going to do 3 really cool experiments. One is the standard pre-post model, right? Or we can do this day in, day out. We're in the process of trying to recruit 100 people, 50 healthy, 50 diseased with anxiety, that we just look at their breath before and after the event, and we see if it changes from an organic perspective. At the same time, we get a sample of their mouth microbiome and their stool microbiome. Cool paper just on its own. The second study comes from a group that's used their system I think it's in Germany. They've put it in movie theaters during specific kinds of movies. So it turns out that when a group of people are watching a comedy versus a horror movie, there's a complete different expression of things that come out of your breath. So this suggests that emotion ties with volatile things that come out of breath. And so we're going to look at the whole room and sample it during phases where no one's in the room, when everyone is listening to a lecture, and then when everyone's doing a specific meditation. I suspect we're going to see crazy mass spec patterns when everyone's meditating in there, right? Because you're going to see the mind creating all these new chemicals that the body is going to exude through this breath. And I think this is how the space is interacting. And we want to get a sense of that. And then we're recruiting 6 people to heavily instrument them. And we're going to put on everything you can imagine on them and get breath at the same time we're measuring all these other things, which would be really cool to see. But it's this arc of we started with looking at the healthy individual, and where it shifted to is really this idea of the human condition is how do you relate to everyone else that's around you, and can that be a power to transform and change who you are?
So going back to your depression and anxiety, because that had a very large population, 250 versus 50.
Yeah.
And you said that towards the end, these people with depression or anxiety disorders, they are looking much more like normal.
Yeah.
So now the question is, if you do a clinical diagnosis of that depression, how do they look like?
Yeah, so we do a lot of surveys, and we do see a resolution of their anxiety and depression, right? So in 7 days, you see them turn in a unique kind of way. We've done studies around PTSD as well, where there's a PTSD specific survey where if you're above a 30 or something score, you have this classic PTSD. And when we administer this survey at the beginning of the week, we tend to recruit people that are in the 50s and 60s in this. So there's no questioning, right? They have it. At the end of the week, 9 out of 10 of these individuals move to this lower than 31, where that's in 7 days, right? There's this complete mind shift that happens. The big question right now then is, how long does this effect last? What's the dosing? What's the effect? What is the patency of it? How long can we actually make it go without someone meditating again? I think it's probably a couple weeks if you stop meditating. Mm-hmm. If you continue to meditate, I think you could sustain it for a couple months, I would imagine.
Mm-hmm. Yeah. And then coming back to your other studies where you're putting GCMS.
Yeah.
That will be really exciting to see. It's just like breathalyzer. You meditate and then figure out what has changed. So you've talked about plasma. You can take plasma and figure out how meditation is changing plasma.
Yep.
And that's where you saw that the plasma is changing in a way that the person is more resilient against virus.
Yeah.
Anything else? So is the plasma is also making the person more resilient against cancer or something else?
Yeah, so
those kind of studies.
Yeah, so we've started doing these adaptive transfer experiments, right? So we can move the plasma onto a cancer cell and show that the energetics of a cancer cell changes. We're starting to do this with C. elegans as well. And so now we're moving from cells to whole organisms to see if we can create some larger kinds of models around aging and disease and other things. And so one of my postdocs has actually created a diabetic model in worms, and they die a little bit earlier. And so the idea now is, could you create resiliency from a diabetic phenotype? We could buy worms that have Alzheimer's, ALS, and could you create some resiliency against that? The other area where we're really expanding, we probably have about 5,000 individuals where we've done a pre and a post microbiome. And so we know there's about 9 or 10 probiotics that go up with meditation, irrespective of disease. And so the idea is, and most of them you can buy these strains that are not toxic, and you could do some biology with that. So C. elegans eat bacteria. And so the simple, stupid thing to do is to take those, feed them to the worms, and see what happens. Is it magical? Could you create combinations of this to then create longevity and other phenotypes? And so we're starting to do all the modeling around that.
So now we have gone plasma, breath, stool. Of course, urine you must have analyzed.
We have lots of urine. We've done tears. We haven't analyzed the tears yet, but
then if you think about it, instead of when you're talking about C. elegans, I was also thinking, what about if the mother, lactating mother, they are meditating? Can they transmit some of the benefits to the baby?
So we have 14, it took forever. It took 2.5 years to collect 14 lactating mothers at the events. There's maybe one every couple events, right? And so we got a breast milk sample before the event started, and then a breast milk sample at the end. We've done the proteomics, we've done metabolomics. We did microbiome, but not much microbiome changes we saw in that. What the proteomics
microbiome in the breast milk.
In the breast milk.
Okay,
that a little bit, not a huge amount. We through metabolomics we see big shifts in sugars, which is what you'd expect, right? Pre to post. The proteomics was quite striking. The pre to post, there's a complete separation. So when you do PCA plots, you get a complete differentiation of pre to post. They don't look anything alike. When we start looking at certain factors, and so I have a lot of these studies come from subjects that come up and want to show something, right? And so the breast milk study, when we ran our first quantum, this woman came up and said, "You need to study this." And it was a bag of breast milk. And I knew exactly what it was because we'd had kids and we pumped a lot of this breast milk. And I don't know if we're ready to do that yet. But then I thought about it. I'm like, we should really launch a study. So about 4 months later, we launched this milk study around this. About 1.5 years into the study, I had a woman come up to me and said, "I got to tell you a really weird story, and maybe this will lead to something, maybe not." She had a nanny that would put breast milk on all the kids' bruises and cuts and stuff. And the wife found out, and she was grossed out by it. She said, "Why are you doing that?" And the nanny explained to her, "Oh, it's got healing properties and stuff." And so they were out and about, and she didn't have any Band-Aids, and her husband had gotten a cut. She rubbed breast milk on her husband's cut, and they noticed some crazy healing properties, right? And so you can do this in a lab really easily. You grow a sheet of dermal fibroblasts, you scratch them, and you see if they fill in. We did this experiment with the breast milk, and the post breast milk creates this super rapid healing. In 48 hours, the entire wound is closed. And it turns out there's proteins that are elevated that predict this injury healing phenotype. I would love to do a study where we follow these kids long-term out in meditating moms and see that they actually have this benefit, right? It would be huge. But it'd be a 10-year plus study to do, which we just don't have the resources to do.
But you just started, so.
That's true. We could do something. But the breast milk does transform. And so we're working on the paper right now around this, and it has crazy changes in lipids and ability to change transcriptome in cells that you treat with and things like that. So we'll see where it goes.
No, I think this is really exciting because you are bringing, I think, when I think of clinical trials, means both you and I know that it's really hard to bring a lot of people to a clinical trial, give the same dosage of intervention. And then measure stuff. And I've been to one of these events, and it's really amazing to see. You can have 1,000 people meditating, and you're right. Every meditation session, the room is completely packed.
And they're all there, right? And so you get really high. And you can tell who's not, because if they're wearing garments, you know what they're doing outside of the room. We can track it.
Yeah, so I think this is really exciting, and we have to see whether these people, when they meditate alone,
Yeah,
are they getting the same benefit. Of course, it will be very hard for people to meditate 35 hours in a week.
right. Yeah,
So I guess this is another example where you have to come to a place like this where you have nothing else to do. Another thing is, it's
right.
almost like they have
Does
nothing
that
else to do.
create a relaxation element,
But
right?
have you studied the spouses or significant others of meditators who are not meditating but are at the
We have. So our early studies have that control that is vacationing, but not meditating.
so just going to the resort
Just they're at the resort, right? And they're drinking their Mai Tais at the pool, maybe doing some light work, those kinds of things. The epigenetic data we did in them is kind of striking, and it makes sense to me. I'm never relaxed on a vacation. I get stressed because you got to
Yeah.
go there and all these things. When we look at their biological age, they actually age a year controls relative to baseline. And so, early suggestion that if you're vacationing, you better vacation the right way, otherwise you're going to get older. Compared to the meditator, we were shocked to see that a new meditator going to an event actually gets a year younger.
Okay, so that also brings up another question. New meditators will see more benefit than the
I think so. I think
meditators, right?
they don't have a preconceived notion of what's going to happen.
Yeah.
Right. So there's more curiosity. When you have more curiosity, I think a lot more neural circuits are forming, and we're starting to see this. So the other big thing we're doing is we're analyzing language, right? So we have these video diary captures that happen at these events, and we're working with a group that can look at the arc of the words that people use over the week. And we were just looking at this data on Friday, and I had to put it in this talk I gave on Sunday or Saturday. And it turns out that the arc that leads from the beginning of the week to the end, where transformation is the main thing that happens, it starts with curiosity. And then what happens on day 2 and 3 is there's a struggle. There's like a conflict, like an inner conflict of, am I doing this right? Why am I here? This is dumb. This is stupid. This is a waste of my time. And then there's this connectedness that they see. And so this is the socialization, which then leads to this transformation in these individuals. And so that arc is this curiosity. And the individuals that are new tend to have that fresher brain, right? They just don't know what to expect.
Yeah. No, that's really exciting because, now as we're discussing, I'm also thinking of too many different ideas, like people on different therapies, how meditation interact with certain therapies.
Yeah.
Because as we know, a drug targets only one molecule, whereas the disease comes from a network.
Yeah.
And whether the effect of meditation on the network can synergize the treatment to improve health.
Right.
So I guess you just started a new field, and I guess just like Joe Dispenza has you as his scientific, I don't know, what do you have a designation there?
No.
Or you're the research guy?
I'm the research guy, but it's independent, right? It's all done. We essentially set up a mobile UCSD when we're there, right? So that's where we want to be in this space, is we're doing independent science.
Yeah.
And so it's at a Joe event, and he is involved intimately in helping us think about ideas and things, but then he steps away. He basically doesn't influence the way things are collected, what happens. It's done in a very scientific way. Everything's blinded. No one, when we get the samples in the lab, they're just a number, right? No one knows what they're from. No one knows how they're stratified. It's not until we're actually analyzing the data where we start to make all these inferences.
Yeah. So I'm also curious whether this has also triggered other meditation camps or other leaders like Joe Dispenza to start similar studies, or are you seeing some of the results that you're finding coming out of other meditation studies?
I don't think anyone's done it at the level that Joe has, but I think people are starting to become curious about it, right? As our papers start to come out, and as I do more of these podcasts and people are starting to see this, that there is this interest in doing this. I think what most of them think is that this can be done it's not a cheap thing to do these kinds of things, right? And we have this nonprofit that funds the lab at a really mega level to be able to do a 1,000-person study. There's no way you could do this with an NIH grant. It's impossible. You'd burn through all 5 years in the first 6 months. And so it's really been amazing to have this support from this nonprofit to think really big and far out about these ideas. But I think there will be an interest in doing this. One of the things that we've been thinking about is not comparing them to people that mimic what Joe does or in that same genre, but can we go back and compare what Joe is doing to all the ancient ways of doing this, right? So there's got to be some correlates to this with plant medicine, which is the hot area in psychiatry right now. Is I think Western medicine is always looking for a pill, something exogenous that you can put in. And plant medicine is hot right now. And there's no argument, right? Psilocybin, ayahuasca, ibogaine, DMT, all of these things do have this ability to change the conceptualization of what the mind does. And they lead to a profound change in anxiety, depression, PTSD, pain, those sorts of things. I think we have to study that. I think we need to create the similar types of data sets that we're creating. Could you look at tears, blood, urine from these individuals in the same arc that you would? The other aspect is breath. I think breath is a unique element of what happens at a week-long event. What happens when you do it in isolation, right? Is there an effect of breath in this? Yoga, transcendental meditation, looking at Buddhist monks who've been doing this for decades of their lives since they were young, kind of thing. And then looking in faith communities, right? Prayer has a very meditative component as well. Does that create some sort of healing modality in individuals when you're invoking God to heal and do these other things? And I think for a lot of this, and I think this is where we're headed with the Joe stuff, it is a mindset, right? And it may be stupid simple as a placebo molecule, but who cares? If we find placebo at the end of this, I'll still be happy. That's a huge discovery. We have a placebo. I don't think it's a single molecule. I think it's a mix of these things, but it's going to lead to some interesting insight ultimately at the end.
But I guess people who are listening to this or seeing this, they will have one simple question. Do they have to just go to one of these events, or can they do it at home, do some regular meditation? May not be 35 hours a week, maybe 30 minutes a day.
Yeah.
And if they do, are they likely to see some benefits? And I guess the answer might be in your study, because those who are meditators who are coming to the event,
Yeah.
is their blood biomarker at the beginning of the event
Different
from or is it similar to the novice who are coming and at the end?
Yeah, so it's similar. So the beginning of an experienced meditator's blood looks a lot like the novice meditator at the end of the week.
Yeah, that's the key.
We see that. And I think we're going to see elements of this. So we're trying to work out the logistics of that experiment that you asked about, right? What do you look at? How do you look at the person that's doing this at home? And so one of the thoughts we have is that there is a way to live stream to these individuals during a week-long event. So could you have a community on site that you're doing the regular kinds of analysis you do in that home environment? At the same time, could you have a community you set up on Zoom that gets to interact with the same number of individuals? You socialize them, but it's a virtual socialization. And then could you have the same number of individuals that are not socializing, but go through the week-long experience, can tune into each of the meditations, the lectures, and then they're living their life while they're trying to do this and see what the effect is. It's going to be graded. I think the people who are at the event, experiencing it, checked out of their lives, where they're not dealing with the stressor of that, are probably going to achieve the most because they have the best setup for it. I think the community that you create on Zoom will likely be the next level. And then I think the individual will see some benefit, but it's not going to be as dramatic as the other two groups. But I think it can be done, right? And then the question becomes, I mean, there are stories that are emerging in his community that show that you don't ever have to come to an event. You could pick up the book, read the book, do some of these meditations, but it's really the focus you put into this. It's that attention and the intensity that you provide into this that leads to that healing element, or not necessarily just healing, but a transformation that you see. And I think this is where most of us struggle, right? Is we get an idea that we're going to do something, and then we do it really well for about 2 weeks, and then life catches up. You're like, I don't know. I'd rather eat burgers and fries because they taste better than salads, kind of thing. And this is the challenge, I think. And I think this is what we have to struggle with. And the mind is the same thing. I think you train your mind to think about good aspects of it for a short amount of time, but then it's a struggle all the time to keep it doing the exact same thing. So
Yeah, so that's we had a pretty long conversation, and I have only one last question.
yeah,
Do you meditate?
I do.
And how long? And because you have had the Kool-Aid.
Yeah.
And means when I went to the event, I also had the Kool-Aid, and I was thinking, okay, so I should go back. And you are right, like it's really hard after you come back, maybe a week or two, and then I kind of fell off the
Yeah.
wagon.
Yeah.
And how do you keep the meditation habit?
So it's,
everybody who is
yeah.
listening,
Yeah.
how do you keep it?
It's hard. So you have to find a community. And so there is a San Diego group that we formed, and they're involved in research on campus that we do around this in a more concerted kind of way, in a deeper kind of way that we just can't do at an event because we have access to fMRIs and our lab and stuff like that. And so we meet every Thursday. And it's one of the things, I can't always go to every Thursday meeting, but it's something I look forward to, right? And it's crazy, like 40, 50, sometimes 60 people show up, and we're meditating as a group, and it's really powerful. I use Joe's meditations once in a while, but I think one of the things that I've learned, I've been to a lot of events, either attending or doing research, and so I'm around all of it. And I think what eventually you learn to do is to have that self-control in sort of your mind, that you can do it in a unique way that doesn't require you to close your eyes, put the headphones on, and listen to that meditation. And so I tend to be very reflective when I go to bed, right? And so we were doing this, we were testing out these patch devices that we have in the lab, and they're amazing for sleep. And Julie, who was doing the analysis for us, she's like, you're in like a gamma state for like the first hour and a half of your sleep. It's weird. And then you just fall asleep. It's bizarre. She's like, do you meditate before you go to bed? I'm like, no. And so then I was thinking about it. I'm like, but I do this like rehearsal, right? This self thing where I think about the day that's happened. I think about the day that's to come. I imagine myself in those scenarios of how I'm going to get through all of those things that are there. And then my brain gets tired and I fall asleep. And so that mental rehearsal is, I think, a form of meditation, and it puts my mind into this really creative, elevated state. And so I think you can do it in very different ways, and it brings you some sort of mind-body connection, I think, ultimately.
Thank you so much, Hemal. This is really exciting. And there are very few scientists on a very different note. There are very few scientists. All of us, we do have a science role, and then we project ourselves as mostly atheist. We don't talk about the religion, faith, or mind, and many people may talk about mind, but not beyond that. And you not only think or act outside the box. Actually, you don't have a box.
That's good.
So this is really refreshing, because many people think of scientists as very cold, insular people.
Yeah.
But I always think that scientists and priests have something in common.
Yeah.
So scientist means, for example, you might have seen the electron microscope image of a mitochondria, but actually beyond that, we have never actually seen the molecule cholesterol, or we haven't seen many things that we describe. So we actually believe in something that is unseen.
Unseen, yeah.
And we strongly believe in that, and we ask people to believe in that.
Yeah.
And priests do the same thing.
Yeah, yeah, for sure.
They haven't seen, and they preach us to believe in the unseen.
Yeah.
So you kind of bring the whole circle back, and it's really gratifying and very eye-opening and mind-opening to grasp all of this together, and to think of, just like we talk about brain-body connection, but actually it's there is something beyond that, mind, and then meditation is a form of exercise of the brain. We can kind of think of that way, and hopefully this type of research will yield, as you said, many of the manuscripts are in preparation, and hopefully in the next 3 to 4 years, we'll begin to see many of the papers coming out. And thank you for your time, because this is the best way we can communicate what we are doing, summarize and synthesize to the public.
Yeah, no, it was
Thank
a
you
lot of
so
fun.
much.
That was great.
Yeah. And thank you, all of you. And, in the last 1 year, this is almost more than a year that we have been doing this Performance Around the Clock podcast. And many viewers have been generous enough to donate. And that is actually helping us to continue this and to have some of the science going on in the lab. Thanks again. And we'll have another episode in a couple of weeks.